Welcome students. Today we are going to discuss oxidative phosphorylation and substrate level phosphorylation. Plants, algae and some bacteria harvest the energy of sunlight through photosynthesis and convert radiant energy into chemical energy. The reduced cellular carbon generated during photosynthesis oxidized to carbon dioxide and water, and this oxidation is coupled to the synthesis of ATP. Respiration takes place in three main stages: glycolysis, the citric acid cycle, and oxidative phosphorylation. The latter comprises the electron transport chain and ATP synthesis. During glycolysis, carbohydrate is converted to pyruvate in the cytosol, and a small amount of ATP is synthesized via substrate level phosphorylation. Pyruvate is subsequently oxidized within the mitochondrial matrix through the citric acid cycle, generating a large amount of reducing equivalents in the form of NADH and FADH2. In the third stage of oxidative phosphorylation, electrons from NADH and FADH2 pass through the electron transport chain in the inner mitochondrial membrane to reduce oxygen. The chemical energy is conserved in the form of an electrochemical proton gradient, which is created by the coupling of electron flow to pumping of proton from the matrix to the intermembrane space. This energy is then converted into chemical energy in the form of ATP by the ATP synthase, also located in the inner membrane that couples ATP synthesis to the flow of protons back into the matrix down their electrochemical gradient. What is phosphorylation? Phosphorylation is the addition of phosphate group to glucose or other organic molecules like proteins or fats to obtain energy in the form of ATP. ATP, the high energy exchange medium in the cell, is synthesized in the mitochondrion by addition of a third phosphate group to ADP in a process referred to as oxidative phosphorylation. ATP is also synthesized by substrate level phosphorylation during glycolysis. ATP is synthesized at the expense of solar energy by photophosphorylation in the chloroplasts of plant cells. Now, what is oxidative phosphorylation? Oxidative phosphorylation is a process in which ATP is formed as a result of transfer of electrons from NADH to FADH2 by a series of electron carriers. The electron transport chain generates no ATP directly. Only 4 of 38 ATP ultimately produced during respiration of glucose are derived from substrate level phosphorylation, 2 from glycolysis and 2 from TCA cycle. The vast majority of the ATP, that is 90%, comes from the energy in the electrons carried by NADH and FADH2. During oxidative phosphorylation, electrons are transferred from electron donors to electron acceptors, such as oxygen, in redox reactions. These redox reactions release energy which is used to form ATP. These redox reactions are carried out by a series of protein complexes within the cell membrane in both prokaryotes and eukaryotes. These proteins are located in the cell's intermembrane space. These linked sets of proteins form the electron transport chain. In eukaryotes, 5 main protein complexes are involved, whereas in prokaryotes, many different enzymes are present using a variety of electron donors and acceptors. The energy released by electrons flowing through this electron transport chain is used to transport protons across the inner mitochondrial membrane in a process called chemoosmosis. This generates potential energy in the form of a pH gradient and an electrical potential across this membrane. This store of energy is tapped by allowing protons to flow back across the membrane and down this gradient through a large enzyme called ATP synthase. This enzyme uses this energy to generate ATP from adenosine diphosphate ADP in a phosphorylation reaction. This reaction is driven by the proton flow, which forces the rotation of a part of the enzyme. The ATP synthase is a rotary mechanical motor. Although oxidative phosphorylation is a vital part of metabolism, it produces reactive oxygen species such as superoxide and hydrogen peroxide, which leads to propagation of free radicals, damaging cells and contributing to disease and possibly aging. The enzymes carrying out this metabolic pathway are also the target of many drugs and poisons that inhibit their activities. Oxidative phosphorylation works by using energy-releasing chemical reactions to drive energy-requiring reactions. The two sets of reactions are said to be coupled. This means one cannot occur without the other. The flow of electrons through the electron transport chain from electron donors such as NADH to electron acceptors such as oxygen is an exergonic process. It releases energy, whereas the synthesis of ATP is an endergonic process which requires an input of energy. Both the electron transport chain and the ATP synthase are embedded in a membrane, and energy is transferred from electron transport chain to the ATP synthase by movements of protons across this membrane in a process called chemoosmosis. In practice, this is like a simple electric circuit, with a current of protons being driven from the negative N side of the membrane to the positive P side by the proton pumping enzymes of the electron transport chain. These enzymes are like a battery and they perform work to drive current through the circuit. The movement of protons creates an electrochemical gradient across the membrane, which is often called the proton motive force. It has two components: a difference in proton concentration, a H positive gradient, and a difference in electric potential, with the N side having a negative charge. ATP synthase releases this stored energy by completing the circuit and allowing protons to flow down the electrochemical gradient back to the N side of the membrane. This kinetic energy drives the rotation of part of the enzyme's structure and couples this motion to the synthesis of ATP. The amount of energy released by oxidative phosphorylation is high compared with the amount produced by anaerobic fermentation. Glycolysis produces only 2 ATP molecules, but somewhere between 30 and 36 ATPs are produced by the oxidative phosphorylation of the 10 NADH and 2 succinate molecules made by converting one molecule of glucose to carbon dioxide and water. While each cycle of beta oxidation of a fatty acid yields about 14 ATPs. These ATP yields are theoretical maximum value. In practice, some protons leak across the membrane, lowering the yield of ATP. Now, we will discuss history of oxidative phosphorylation. The field of oxidative phosphorylation began with the report in 1906 by Arthur Haddon of a vital role for phosphate in cellular fermentation. But initially, only sugar phosphates were known to be involved. However, in the early 1940s, the link between the oxidation of sugar and the generation of ATP was firmly established by Hermann Kelka, confirming the central role of ATP in energy transfer that had been proposed by Ritz Albert Lippmann in 1941. Later in 1949, Maurice Redikin and Albert L. Leininger proved that the coenzyme NADH linked metabolic pathways such as the citric acid cycle and the synthesis of ATP. For another 20 years, the mechanism by which ATP is generated remained mysterious, with scientists searching for an elusive high-energy intermediate that would link oxidation and phosphorylation reactions. This puzzle was solved by Peter D. Michel with the publication of the chemoosmotic theory in 1961. At first, this proposal was highly controversial, but it was slowly accepted and Michel was awarded a Nobel Prize in 1978. Subsequently, research concentrated on purifying and characterizing the enzymes involved, with major contributions being made by David E. Green on the complexes of the electron transport chain, as well as Freeman Racker on the ATP synthase. A critical step towards solving the mechanism of the ATP synthase was provided by Paul D. Boyer by his development in 1973 of the binding change mechanism, followed by his radical proposal of rotational catalysis in 1982. More recent work has included structural studies on the enzymes involved in oxidative phosphorylation by John E. Walker, with Walker and Boyer being awarded a Nobel Prize in 1997. Now, we will discuss mechanism of oxidative phosphorylation. Three important theories have been proposed to explain the mechanism of oxidative phosphorylation. These theories explain how the energy transfer between electron transport and ATP synthesis takes place. These are chemical coupling hypothesis, conformational coupling hypothesis, and the chemoosmotic hypothesis. Now, we will discuss them one by one. Number one is chemical coupling hypothesis. It was first proposed by Slater in 1953 and is based on the principles of substrate level phosphorylation. The hypothesis postulates that a high energy intermediate is produced as electrons are passed from one carrier to the next. However, no such high energy intermediates have been shown to exist, and the need for intact mitochondrial membranes for effective oxidative phosphorylation is not explained by this hypothesis. For the explanation of hypothesis, it is proposed that two hypothetical coupling factors called X and E are involved at each ATP generating step. It is further proposed that coupling factors required at 3 ATP generating steps are different. They are designated as X1 and E1, X2 and E2, and X3 and E3. The various steps of mechanism are as: first, the coupling factor X first combines with the respiratory enzyme like cytochrome B to form a high energy intermediate complex. Second, the high energy intermediate complex combines with phosphate to form phosphorylated intermediate containing a high energy phosphate group. At this step, the respiratory enzyme is removed. Now third, the phosphorylated intermediate now combines with another coupling factor E to replace first coupling factor X and to form energy rich phosphorylated complex, which catalyzes the synthesis of ATP from ADP and regeneration of the coupling factor E. Thus, it is presumed that the function of coupling factors X and E is to transfer the energy released in redox reactions for ATP synthesis. Number two is conformational coupling hypothesis. It was first proposed by Boyer in 1964. According to this hypothesis, the energy produced during electron transfer is converted by conformational changes in the molecules comprising the mitochondrial membrane and matrix, which may be the driving force for ATP formation. The main conformational changes have been observed in ATP synthetase particles and crusty of mitochondria. The crusty assume different forms during different functional stages of the mitochondria. Similarly, the ATP synthetase particles also assume different shapes like disc, dumbbell, or spherical when there is lack of energy supply and the mitochondria is in non-energized state. The crusty are in the form of straight flattened sacs, and stock of ATP synthetase particles are called repeating unities. Becomes contracted and its headpiece or F1 part becomes flattened to form a disc. When isolated mitochondria are supplied with substrates or kept in solution containing ATP, they are converted into energized state. At this stage, the crustacea become more organized and assume vesicular form, whereas the stems of ATP synthetase particles assume an extended form and F1 part becomes dumbbell-shaped. When only inorganic phosphate and no ADP is supplied to mitochondria, the membranes of the crustacea become convoluted, assuming zigzag shape, whereas the stem of the ATP synthetase particles become elongated and F1 part becomes spherical. When ADP is added, the energy twisted state is changed to energized state, which can be converted to non-energized state by addition of such cations or uncouplers that can be transported across the membrane. Of the above conformational changes, the twisted form stores the energy released during electron transport. When energy is added for ATP synthesis, the energy twisted or energized state returns to stable non-energized state, releasing energy which is utilized for the synthesis of ATP from ADP and inorganic phosphate. Boyer in 1965 proposed that there is a direct communication between electron transfer catalysts and ATP synthesizing components through polypeptide-polypeptide interaction. Boyer and Slater 1974 proposed a modified conformational coupling hypothesis, which postulates that electron transfer induces conformational changes leading to translocation of protons. Conformational changes in electron transfer proteins induce changes in ATP synthesizing protein components. They believe that passage of protons through F1 can change the conformation of its protein, and such proton-induced conformational changes near the active site can synthesize ATP. ADP and inorganic phosphate can combine spontaneously to form ATP in the active site of F1 of ATPase without requiring free energy. ATP formed is tightly bound ATPase. The energy is therefore required to release tightly bound ATP molecules from ATPase. The protons, when bind elsewhere other than the active site, can cause conformational changes in F1 part of ATPase, resulting in to release of ATP. The protons are released into the solution and M side of the membrane. Now, number 3 is the chemoosmotic hypothesis. It was proposed by British biochemist Peter Mitchell in 1961. This theory is most convincing and acceptable to date. According to the chemoosmotic theory, the orientation of electron carriers within the mitochondrial inner membrane allows for the transfer of protons across the inner membrane during electron flow. Because the inner mitochondrial membrane is impermeable to H positive and electrochemical proton gradient can build up. As the proton concentration in the intermembrane space rises above that in the matrix becomes slightly negative in charge. This internal negativity attracts the positively charged protons and induces them to re-enter the matrix. The higher outer concentration tends to drive protons back in by diffusion. Since membranes are relatively impermeable to ions, most of the protons that re-enter the matrix pass through special proton channels in the inner mitochondrial membrane. When the protons pass through these channels, synthesize ATP from ADP and inorganic phosphate within the matrix. The ATP is then transported by facilitated diffusion out of the mitochondria and into the cell's cytoplasm. Because the chemical formation of ATP is driven by a diffusion force similar to osmosis, this process is referred to as chemoosmosis. Thus, the electron transport chain uses electrons harvested in aerobic respiration to pump a large number of protons across the inner mitochondrial membrane. Their subsequent re-entry into the mitochondrial matrix drives the synthesis of ATP by chemoosmosis. The chemoosmotic model suggests that one ATP molecule is generated for each proton pump activated by the electron transport chain. As the electron from NADH activates 3 pumps and those from FADH2 activates 2. Thus, each molecule of NADH and FADH